It's my great pleasure to introduce our speaker today, Associate Professor Roseanne Quinnell. Roseanne's from Life and Environmental Sciences in the Faculty of Science. Her research and teaching focus on plant sciences and the use of technology-enhanced solutions to improve student learning. And some examples of this work include botany, zoology, and human biology viral microscopy slide collections, the eBot Botanical Image Repository, Electron Laboratory Notebooks, and the University Campus Flora apps. Today, Roseanne is discussing Botanische Wandtafeln, a series of technical scientific illustrations produced between 1874 and 1911 and distributed globally as teaching tools to support student learning in botany. Reliance on these types of materials has fallen out of favour for various reasons, which Roseanne will discuss, and she'll explain how reutilising this series in a digital platform allows for enriched dialogue about how science is communicated. So please join me in welcoming Roseanne.
Thank you. Thank you, what a fantastic introduction. I don't get that. I would like you to come to all my lectures, please, and give me that same courtesy of introducing me. So first and foremost, I too would like to pay my respects to the Gadigal people of the Eora Nation, the land upon the land where we're meeting today. We've got a long history of observation in botanical science, connecting us through from the Gadigal people all the way through to present day. So plants connect us to the landscape in interesting ways, and I am unashamedly a botanist, and I can see that there are some loud and proud botanists in the audience today. So the title of this talk is Illustrations to Micrographs. So when you communicate anything in science, you can use words. Words are great, but other times illustration is really, really important. And when you find illustrations of the quality of these illustrations that have got value not only biologically, but also historically, they're such a joy to work with, and it means that you can include narratives with your students, not just about the biology, but also about the people and the history of biological observation. And I'll just go back one. So part of my job as an educator in botany is to assist students to be able to recognise the patterns in biology, particularly in botany. So that's what we do. As scientists, we communicate biological patterns in words and in pictures. And there are conventions where we're kind of constrained by the conventions of how we draw things as we are with how we write things. Our descriptions are actually as constrained as our illustrations. And I'm very pleased that I've got a couple of my botany students in the audience too. So I gave a tutorial earlier this week, so one of the slides from that tutorial will pop up. So we still teach the same way, introducing students to the conventions of our discipline in how we write and how we record our observations. And when we're recording our observations, scales are absolutely crucial. And there's a couple of scales here. There's spatial scale, size, and there's also temporal scale. In biology, importantly, it's developmental stages, but there's also temporal scales, of course, in the timeline of evolution. So temporal scale is critical to our understanding of biology, and scale is also critical in understanding the size of things. So we need to be able to engage with both of those elements in biological systems. So let's cut to the chase and talk about this bloke, Kanai. I will probably default to using his name just as Nye and make the K silent, so I'm going to anglicise that, but just pretend you're hearing it at Kanai. Leopold Kanai, he was really Polish, but he did most of his studies in Germany, and he created these illustrations. So a very patient man, I would say, but don't forget, in those days, time was abundant. Time now is super pressured. So this is Leopold Nye. He created these. These scientific illustrations were created in about 1911. The first run of these illustrations were given to the Kew Gardens. So Kew Gardens has got the first print of these, which is also quite fascinating. So he was corresponding with the head of Kew Gardens at the time, and through those correspondences, just before World War I, he was able to take the first printings of these wall charts off to Kew Gardens, where they're curated as carefully as they are in our own museum. So they were distributed globally. They were still distributed globally up until the 1960s, which is, for some of us is like real lifetime. For others of you, you may only dream of what happened in the 60s, but some of us have got a memory. Well, I don't really have a memory of that. I was a wee baby in 1960. And when I say globally, I mean globally. We have our collection in Australia. There were collections in the USA, in the Netherlands, and in Ireland. And so in all of those places, people are starting to do what we've done, is digitise those collections to preserve them. The most comprehensive collection of Nye's wall charts is in the Netherlands, and so you can see those and download them pretty readily. They've got all 120 of these teaching charts. We've got 30 at the moment in our collection, but talking to Julie earlier today, we may have some other ones that we can digitise to add so we can get as close as possible to this kind of digital collection. And for me, we're starting to reprint them and laminate them, so we've got them in our lab classes. So students can touch them because they're a copy and it just makes them available. It also means that they're available to students if they see a real corker that they want to take home. They can log into the Library Digital Archive, as can all of you, download one, print it off, laminate it, and then have it as art in your home. Because this is where we're sitting, really. We're sitting between science and art when we're looking at these images over here. You don't have to be a botanist or a mycologist or somebody who studies algae to appreciate the beauty that's in these posters. So this is one of the posters we have in our collection. So this is a lichen. Within the lichen, a lichen is a symbiosis between a fungus and an algae, and here's a cyanobacterial algae that's in the association with the fungus to form this lichen. And I found this really, I think, quite good paper. Well, it's in a book, The Nature and Classification of Lichens, Lichen and its Algal Host. And when I was reading through this, it cites Nye's research. So Kenai was researching into lichen. That was one of his favourite class of organisms to research. So I found this citation, and then it's down here, of course in German, which was the language of the day. German was the language of science up until quite recently. So if you're a scientist at the turn of last century, you would be writing papers in German. So English is a relatively recent language to be communicating science. Oftentimes it was in German. So this is Nye's work, and here is his educational plate. So here we've got this kind of segue between Nye's research and an educational tool. In modern pedagogy, we will call that research-informed teaching or research-led teaching. So bloody old news that, the old research-informed teaching, not new information. It was normal practice over 100 years ago. And these wall charts were really important to be able to teach key concepts in botany. So if you look carefully at these two flowers, they are from the same species, but there's a dimorphism. So there's two morphological types in this flower. And if you look closely, you'll see here that the female part, the gynoecium here, has a very short style and stigmatic surface. And the gynoecium over here has got a very long style and stigmatic surface. And the anthers here are just at the neck of the flowers, and then the anthers here are further down. So these sorts of, the key concept here is that you don't want the flower, the plant wants, and I'm being deliberately anthropomorphic, is avoiding self-pollination. So if a pollinator comes along here, dips its parts of its probably head. I'm not a zoologist. parts of its head or something that's got a long thingy to be able... I can tell there's zoologists in the audience though. They think this is a crack up. Into the flower, then the pollen is not going to be able to be transferred to a similar flower. It's going to only be transferred to a different sort of flower. So it's avoiding self-pollination. So that's a key concept. Not all plants do this, not all flowers do this. Sometimes there's a temporal difference between when the pollen is mature and when the stigmatic surface becomes available or receptive to pollen. So plants have got different ways of being able to restrict how much self-pollination is incurred. So these are longitudinal sections through the flower. Students do this in my prac classes quite a lot. They do longitudinal sections. They're looking at the positions of the anther, whether these parts are connected or free from the other parts in the flower structure. They're looking at whether this gynoecium, the female part, is obvious or whether it's submerged into the base of the flower, so whether the ovary is superior or inferior. So there's a whole lot of training in learning how to see like a botanist that you can teach or consolidate using these sorts of images, which is why I'm still using them in my classes. So this was looking at the heterostyle of this particular plant. The other thing to note, and I'll reiterate this a little bit later on, is that this illustration is at different scales. So this scale is not the same as this scale. So this is the blow up of this style here. This is the blow up of the top of the style here. And so on and so forth through the plant. And then I found this other paper that was, or it's actually, when I say paper, I mean blog, because everything's online now, right? So this is from 2015, and it was a blog that was using one of Nye's illustrations, and it's you don't really see a plant until you draw it. Now we encourage our students now to draw, but drawing is absolutely a different activity than taking a photograph. It requires a higher order of observation to be able to declare exactly where you see the positions of these xylems in here, or even the ratio of this stele, and I'm using botanical terms because I can't help it, to the rest of the transverse section of a root. So very, very careful observation is required before you declare where you're going to put your pencil when you're doing your drawing. You don't have to think much to take a photograph, which means you don't have to observe carefully to take a photograph. So there's a different level of engagement when you're drawing and looking than when you're taking a photograph. So what we gain in photography, we lose in that metacognition of being able to observe very carefully. And it's another reason to use these wall charts in teaching. It alerts students to that if you had to draw what you're seeing down the microscope, how careful would you need to be, not only to draw it as an aide-memoire for yourself, but also so another scientist could look at your drawing to be able to understand what you were seeing down the microscope. And this is where we get to the slide that I put up in the tutorial on Monday. So the left is a photograph down a microscope, so it's a photomicrograph. And on the right-hand side is a map diagram, a legitimate scientific rendering of the image on the left. So a map diagram is really a tissue map diagram. And up until about 10 years ago, we used to assess students' ability to draw exactly in this way, and we still encourage students to draw in this kind of shorthand way. So if you see on this slide on the left, there's a lot of cells in here. You see that those cells are absent in the map diagram, but there is a label that's very descriptive, cortical parenchyma. Kind of means that every cell that you see in this region just looks like a parenchyma cell. So you don't need to draw it. You just need to declare what sort of cell type you think it is and the region in the transverse section. Again, this is of a root. So it's kind of a shorthand way of drawing this photograph or drawing it from when you're looking down a microscope. So drawing a photograph is like a redundant exercise. Drawing your observation down a microscope is a really useful exercise. So this is what we do in our botany class. And of course, you can't really label anything unless when you're sitting at your microscope, you're rendering your drawing not at one magnification, you're constantly shifting between 4 times, 10 times and 40 times. So you're shifting the spatial scale of what you're looking at. So this photograph becomes a consensus image of 3 magnifications, whereas this photograph is only a snapshot taken at 1 magnification. So again, there's a limitation at the level of detail that you can capture in that photograph, and also focal plane. So a photograph is a rendering of one focal plane, whereas a drawing, because you can use the microscope to do a little Z refocus, you can then consolidate what you've seen in that drawing and it becomes actually a better rendering of what you've seen. It's the conventions of illustration in botany. And I also know there's a few people who did botany at this university a couple of years ago. I can see them maybe 3 or 4 years ago who are going, yes, we understood all that. I'm going to take you back to the context of these posters at this university. So what I think, and I'm just speculating, is who brought these posters to Australia or who ordered these posters to decorate the walls of our laboratory classes in botany. And I'm speculating that it was likely to be A.A. Lawson. So his fantastic name, Abercrombie and Struther Lawson. He was Scottish via Canada into Australia, and he was our first professor of botany. And he started in 1913 and he finished up actually when he passed away in 1920. And he studied under Bauer in Glasgow, and the tradition of teaching botany was very much focusing on morphological characteristics and understanding the connection between form and function. When he started, there were only 136 students in botany. 10 years later, after 10 years, there had been 3,000 students coming through botany. So botany was pretty popular back in the day. I wish it was as popular now. I'm desperately fighting to make sure that it is popular, because it's absolutely fundamental to actually the air we breathe, if nothing else. So A.A. Lawson also was very much involved in giving public lectures in botany. So that was one of his things. He used to want to take botany to the people. So he's arguably one of our first champions of botanical literacy, to champion botanical literacy of the society more broadly. And we've got a few people like me who are still trying to champion that notion of botanical literacy for everybody, particularly on our campus. He was also responsible for the botany building. So if you ever look at the Macleay building quite carefully, you can see there's a little add-on at the north end. That's the botany building. And that was completed just before he passed away. I think it was opened in 1926. And so we've been teaching botany in that venue for 92 to 93 years. And again, sadly, that's going to come to a close. I believe we won't be teaching in that venue next year, which fills me with dread as to how to manage to teach without that. I don't know, it's a beautiful building. It's a great place to teach and learn. So from his an article when he passed away, I really liked this. He believed that unless you could draw the living organisms and the changes that took place during life and the reproductive stages, you do not understand the subject. He was also a really strong advocate for having hands-on teaching. So teaching in the practical situation, teaching in the field, was a really strong legacy that started with him, and it continues through all of our science teaching today, particularly in biology. So yeah, he was a good egg. He had his own versions of how to publicly display the organisms, and he used these things, lantern slides. So they're, I don't know if they're a glass slide, you put a lantern behind them and the images get projected. So he hand coloured his lantern slides, and that collection is in the Macleay Museum. And in 2016, I worked with an advanced student and we did like a historical perspective on that. We got it published in the Muse. So 2016 was a really important year for botany because it was 100 years of the Botanic Gardens. So significant connection to botany at this university. So like I've tried to get through to you that this drawing is a really interesting drawing because it goes from the macro scale. So this is what this is up here. I've found these images on the net. And then it progressively does a deeper dive into the cellular. So this would have been drawn probably just by eye. And then from here on in, a microscope would have been used to render these other illustrations. And of course, if you look down a microscope, you can see very similar structures. This is the cyanobacteria. There's another cyanobacteria. And these, because I can't help myself but tell you about it, it's so hardwired. These heterocysts are nitrogen-fixing cells in this multicellular cyanobacterium. So again, a critical process on the planet to be able to fix dinitrogen from the atmosphere into usable biological elements. So I guess the image does what the photograph can't do. It kind of unclutters it from unfocused material and it focuses your eye in on the important parts. So if a student was to look at down the microscope at this, they should pretty easily be able to see where you are, through the illustration. All these mini illustrations, and there's 14 little mini illustrations here, would have come with a description for each of those illustrations, which we don't have, but I'm sure if we find them online, we'll be able to add them to the digital collections so that they become even more useful in the descriptive text that goes with these images. And I did note that I was introduced with some other work I've been doing. So this is one of our wall charts that we've got. This is a liverwort. Here's one that we don't have, but it's still in the Nye collection. This is a liverwort. It's a so-called primitive plant. And we still look at these sorts of slides today. We look at the real images. We look at slides made from plants like this. And we now have a series of virtual microscope slides. And I'm going to attempt to get it to launch. So technology can be really good. But the limitation here is when you draw through one of these little pores in the surface of the liverwort, you can kind of get a 3D image. You can draw it in the third dimension that you can't do when you've taken a slice through these pores. You can only get it in the 2 dimensions. So in how these slides are made, you're not getting a full picture of what that structure really looks like. I think we're all right. So in my opinion, and I think in the opinions of many other people around the world who have been using these sorts of illustrations essentially for more than 100 years, these are the exemplars of best practice in careful observation and communication of key concepts through illustrations. And the posters from our own collection have been digitised and reprinted, and you can get, and we're also using other ones. So I don't know if you're familiar with other people like Haeckel, who did a whole load of diatoms. So it was a bit of a Victorian fashion, was to assemble your diatoms, all different shapes and sizes. I know. People had so much time in the olden days to be able to create works of art from this, and Haeckel did some gorgeous illustrations. I also found this online because you can find these posters not only in kind of legitimate collections, and I'm not saying that Pinterest is not a legitimate collection, but you can find all these illustrations, fantastic botanical illustrations on Pinterest. And I found this one and I went, holy moly, look at that. That's US. Oh no, it is Australian dollars. Phew. It's like, so these are really valuable. If nothing else, they've got a monetary value. And that's pretty well all I want to say. And I'm really sorry that I've kept you from looking at these beautiful illustrations. So when we finish with questions, if there's any questions, then we can go off and have a look at those beautiful posters. Oh, perfect timing.
How much time does it take approximately to make like a diagram underneath the microscope, like a good one?
For me, I could probably do it in 10 minutes, a reasonable one. To really finesse it, it could take me another day. But I could get enough information in a 10-minute diagram to be able to convey to another scientist, and particularly another botanist, as to what I was seeing under the microscope. Yeah, so that's not a map diagram.
No.
Yeah.
But when do you think that shift happened?
I don't know. I think it's a utility. The question is, when did we shift away from doing map diagrams, from doing cell diagrams to doing map diagrams? And I think it's depending on what you're going to use the diagram for. So if you want to really convey a lot of detail and it's more of an educational context, or it's a new species that you really want to defend, I think more detail the better. But if it's in a class assessment, or if it's, you just want to do a diagram to confirm, so if I saw something and I wanted to show you, but I knew the slide wouldn't last, I'd do a diagram of it and then I'd be telling you about that when we met up. But I think that they were both legitimate up until the age of photography. And photography meant that people knew that they could capture an image very, very quickly down a microscope. But it's also the technology of being able to do the right attachment. So cameras had to be little for a start. So you'd have to have a camera that was little enough to be able to attach to a microscope to be able to take those photographs. So I think you'd have to go back through, the history of the technology to see when it became a mass movement from moving away from doing cells to doing map diagrams.
Thank you, Roseanne, and please come up.
Yes, please.